start with a 10 to 15 minute baseline. We record neural activity, give random rewards, and use 2 selected neurons to calibrate the task difficulty, so roughly 30% of trials end in a success or failure by chance. The animal then undergoes a 60-minute testing session, with each trial ending in a success, failure, or timeout, followed by roughly 10 seconds between the trials to allow the animal to obtain a reward if necessary. Finally, you can see here the experimental setup with the animal in the cage, the connected electrodes, and the sugar solution dispenser for providing a reward right here. In order to assess the animal's location in the cage and subsequently its motivation for reward, we recorded videos like this of the animal's position in the cage across all testing sessions. So while we were just beginning data collection, we have successfully set up the neurofeedback task and tested our pilot animals. So today I'm going to share with you our validation results and preliminary findings through a series of 5 questions. The first question is whether we can record single neuron spiking activity in real time. And as you can see here, the answer is yes. We were able to isolate individual neural activity from a number of different channels simultaneously. The second question is whether we can use this neural activity to control the frequency of a sound played to the animal. To do this, we define the cursor value, which is the difference between the smooth spike trains of the up and down units, which again increase and decrease the auditory frequency respectively. We set limits on the rewarded and unrewarded targets, and then map the cursor value to the auditory frequency played using a second order polynomial graphed here on the left. In addition, in order to ensure the cursor values used in real time are the same as the ones used in offline analysis, we confirm that the online cursor values graphed in blue, which were calculated in real time, are the same as the offline cursor values obtained by back calculating the cursor value from the required trains, which we later analyzed, with an R-squared value between the curves of 0.94. Next, we ask whether neural firing rate does increase for the up unit or decrease for the down unit, since the goal is to train animals to increase the cursor value. Using a z-scored firing rate, we saw that in time immediately before success, there's a sharp increase in the up unit firing rate and a slight dip in the down unit firing rate as expected. To better quantify this, we define the modulation depth, which is the average firing rate of a neuron in the 1 second window before success minus the average firing rate in the 1 second window after success. With this metric, we see that the up neuron has a very positive modulation depth that is significantly greater than 0, while the down neuron has a slightly negative modulation depth as expected. The fourth question is whether there are any early signs of learning. Comparing the modulation depth from the first 3 days of testing to the last 3 days of testing on our pilot animal, we see that the modulation depth increases for the up neuron and decreases for the down neuron, indicating that the animal does learn to control its neural activity. We saw further evidence of this in the histogram of auditory frequencies, where the mean frequency increased slightly but significantly between the first 3 days of testing to the last 3 days, with a p-value of less than 0.01. Finally, there was evidence of learning within the sessions over the last 3 days, where the number of events per minute for rewarded targets increased from the first 10 minutes of testing to the last 10 minutes of testing, while the number of events per minute for rewarded targets decreased over the same time period. Lastly, we want to ask whether we could track the animal's location and use it to gain insights into the motivational state of the animal. As seen in this video, we were able to successfully extract the position of various body parts by using the open source DeepLab-Cut software, which uses machine learning to train deep neural networks in order to extract and follow animal location. Using this data, we're able to extract information about the animal's behavior during the task. For example, we compared the mean nose distance from the sucrose solution dispenser following a success on the first 3 days versus the last 3 days. With this, we can see the animal was much more motivated for a reward in the last 3 days as compared to the first 3 days, as also can be seen in its mean time to retrieve its reward being much lower in the later days. So in summary, we showed that 1, we can record single unit neural activity in real time and use it to control the frequency of a sound through neurofeedback. 2, we can train animals to control single neurons in the hippocampus. And 3, we can determine animal behavior and motivation by accurately tracking position. For next steps, we are currently testing our first 2 animals. And once we obtain sufficient data on all 10 animals, we will assess how activity in the striatum changes during learning. So why should you care? There are many reasons, but I'll let you know about one application. With a rapidly aging population, dementia and memory loss are increasingly prominent burdens on our global healthcare system, affecting roughly 50 million people worldwide each year, with no current medications or treatments able to fully slow or stop its progression. A novel approach to investigate this is to use deep brain stimulation, where electrodes are implanted into the brain to deliver current to a targeted region. However, this technique has been largely ineffective because memory networks in the brain are very intricate, and this approach, because of its core stimulation, bombards entire memory networks with synchronous stimulation, disrupting these memory circuits. On the other hand, a neurofeedback approach, such as the one demonstrated in our study, offers a potential closed-loop approach to modulate the activity of individual neurons and very precisely control memory networks in the brain without requiring stimulation, to not only improve memory, but also, and more importantly, understand how these memory circuits work. So these are my references, and I would like to thank you all very much for listening and give a special thank you to Creme, Dr. Valiante, and the entire Non2Brain Laboratory for their support, as well as to UNIR for organizing the event and the University of Toronto for sponsoring my summer research, and I'd be happy to take any questions.
Thank you, Michael, for your presentation. Now I would like to address the judges and ask if you have any questions. So judges, you can please unmute yourself and then ask your questions to Michael.
Hi, Michael. That was a great presentation. I really enjoyed it. With regards to your applications with dementia, what would be some of your clinical hurdles to moving into that application and demonstrating the feasibility?
Yeah, for sure. I think the major, I can just go back to that slide, but I think the major application is in when you're treating patients, you mainly have to focus on studying epilepsy patients. So because when you're doing these studies and you're doing them in humans, you do require to have electrodes implanted into the brain. It's often not practical to do this research on, I guess, people who don't already need to have the electrodes implanted. So I guess the limitation is there have been some differences shown between people's brains from patients with epilepsy compared to patients without epilepsy. So I think that's a major clinical hurdle in terms of getting over that. But that said, there are many investigating implanting electrodes into the brain for memory loss. So it is there in the clinical side, but it's just there is that hurdle of actually having to implant those electrodes.
Great, thank you so much.
Hi, Michael. Great presentation. Thank you for that. Very interesting. I just had a follow-up question, I guess, to that. You spoke about the differences between the humans and the rodents, but then what are the similarities that would kind of help your research saying that, you know, what you found in the rodent can be translatable in the human?
Yeah, so I think the thing with like in implementing these technologies is we know that in humans, this hippocampal single neuron learning can occur, but I guess the question is like, how does it occur? And understanding whether the network that we believe facilitates this learning is actually the one that does it. And if we understand the interplay between those two systems, it would help us better understand how we could target certain regions of the brain to help treat memory loss and dementia and stuff like that. So just using the rodent model to just understand those circuits.
Okay, so we are now going to move on to the next presenter. Thank you, Michael, again. Raymond, are you ready to present?
Hi, yes.